Hey there! So today our chapter which we are gonna do is how do organisms reproduce, class 10. It's a biology chapter and it is very easy. Okay, so here's the overview of the chapter which we will be doing through a series of videos. That is the introduction to reproduction and asexual reproduction, in which we have 6 types: the binary and the multiple fission, budding, fragmentation, spore formation, regeneration, vegetative propagation, and then comes the sexual reproduction, which we are going to do in 2 ways. One is in flowering plants, and the second one is in human beings, that is male and female. Coming to the next topic is reproductive health, in which there are 2 categories: sexually transmitted diseases and contraceptive methods. And then comes the summary. That means we move towards the end of the chapter. Now, for introduction, what is reproduction? Well, reproduction is a biological process by which new individuals of the same species are produced by the existing organisms. And as a matter of fact, reproduction can be asexual as well as sexual. Now, when I say asexual, it basically involves one parent. On the other hand, sexual involves 2 parents. Of course, we'll go into greater detail of both of them in the next few videos. Next, why is reproduction essential? Now, though reproduction is not an essential life process for an individual, it is very necessary for the perpetuation of the species. Now, perpetuation means the survival of the species on the earth. Reproduction is not considered a life process because it is not important for the survival of an individual, but it is important for the survival of the species for the generations to come. Okay? I hope that's clear. And now moving on to asexual reproduction, then there are 6 types of asexual reproductions which are displayed right in front of you. That includes fission, fragmentation, budding, vegetative propagation, regeneration, and spore formation. We will go into greater detail into each of the topic as we move ahead with a series of videos. Now, the first part is fission. So fission is of 2 types, the binary fission and the multiple fission. First we are going to do binary fission. So binary fission. Binary fission is used by bacteria, we can say most probably in bacteria, an organism whose cells do not contain a nucleus copy. They then divide into 2 identical organisms. Example, bacteria, which is mentioned above. And here you can see a picture. Let me show it to you. This is the picture, right? So here, this involves the chromatic material over here, which you guys can see, right? Slowly, as it reaches into the stage of reproduction, the chromatic material gets divided. And as the chromatic material gets divided, slowly the nucleus, the cytoplasm, and the entire organism as a whole starts dividing. After this is done, you obtain 2 new daughter cells. Okay? Moving on to the binary fission in amoeba. I'm sure most of you must have heard about amoeba in your smaller grades. Yes, and you must have done this as well, but we'll go into greater detail this time. So in binary fission, this applies for all organisms doing binary fission. So the binary fission, the parent cell divides into 2 after reaching a point where it has fully grown. So when the parent cell is in the stage of reproduction, it divides into 2 daughter cells. So as you guys can see here, this is an amoeba. It's a parent cell, right? It further, slowly, first the nucleus lengthens and divides into 2. Here you can see it's dividing into 2, and then slowly the cytoplasm divides into 2, and then you obtain the 2 daughter cells. The next slide. Coming to binary fission, leishmania. Now leishmania, over here you can see it's an organism quite wobbly and with a flagella structure over here, right? Okay. So here you can see in this leishmania that you're having another flagella structure developing. And then you see that there are two nucleuses and the flagella structure is growing even more. Soon the nucleus split and then the cytoplasm of leishmania also splits like this. Now here I would like to mention, let's go back to the previous slide, and here you can see amoeba can divide into like two daughter cells from any direction. Let me just show you. For example, amoeba can divide like this. Yeah, it can divide like this, or like this, or like this. It can divide in any direction. But is it the same for leishmania? Well, I don't think so. Come, let's see. Okay, so for leishmania, it is not the same. Why do I say so? Okay, so now here's the structure for leishmania, as you guys can see. So leishmania, right? So over here, you can see that leishmania only divides from one particular direction. That's because leishmania is bilaterally symmetrical, because of which it divides only from one portion, which I'm highlighting over here using the spotlight. I hope it's clear. Let's move ahead. Okay, moving on to multiple fission. The parent cell in multiple fission splits to form many new organisms. A very great example which you have also learned in previous classes is plasmodium. Yes, you heard it right. Plasmodium, I'm sure you must have read in your previous classes, is the organism that causes malaria, right? Okay, so this is the plasmodium. As you can see, this is the nucleus of the plasmodium, this is the cell, and this is the cyst wall. Now, plasmodium has a cyst wall surrounding it when it reaches to a reproductive state. Then, as you guys can see, the multiple fusion in plasmodium takes place, and you can see that the nucleus divides into many little organisms. These are all organisms going to develop into new organisms. And let me tell you that this divides under unfavorable conditions. You can still see the cyst walls, right? Now, this division takes place in unfavorable conditions, and when the conditions are favorable, then the cyst wall breaks and the daughter cells are released out of the plasmodium. Okay, so binary fusion versus multiple fusion. Whoa, now this is going to be a great fight. Okay, so here are the differences on binary fusion and multiple fusion. Let me give them a brief on them. Binary fusion. So in this, the nucleus divides into two daughter nuclei, just as I had shown before for amoeba and for bacteria, that the nucleus is dividing into two daughter nuclei. Whereas for multiple fusion, nucleus divides into many daughter nuclei, just like how I had shown for plasmodium. Now, for binary fusion, constriction is formed in the cell after nuclear division. That means that there is a constriction in the cell after the division. And for multiple fusion, division of cytoplasm occurs very late. Thus, many cells are produced simultaneously. So in this, we can say that in binary fusion, the division of cytoplasm takes place at early stages, like along with the nucleus. But on the other hand, for multiple fusion, the division of binary fusion the division of cytoplasm takes really late. Okay. Now, the third point is only two daughter cells are produced in binary fusion. On the other hand, in multiple fusion, many daughter cells are produced at one time. Then binary fission takes place in favorable conditions, and on the other hand, multiple fission takes place in adverse conditions. So you must be having that question that I had mentioned before, that the cyst wall breaks in favorable conditions. But let me clarify this, that the division for multiple fission takes place in adverse, that means unfavorable conditions. On the other hand, the division of of amoeba or any organism performing binary fission only takes place under favorable conditions. So I hope that this lesson, this topic of the lesson was clear enough. If not, then please feel free to go back and watch the video once again. Thank you and have a nice day.